The second set of reactions that make up photosynthesis go by several different names. They represent the synthesis part of photosynthesis. Sometimes they are called the light-independent reactions or the dark reactions because they do not directly require light. Sometimes they are also called the Calvin-Benson cycle or sometimes just the good old Calvin cycle. So what's the point of this set of reactions? Well, of course, it's to make glucose. We're going to make sugars. That's what we're synthesizing in these reactions. Now, making glucose, building a big molecule, is endergonic in nature. It's anabolic in nature, so that requires energy. Where do we get the energy? Well, we get that energy from the ATP and the NADPH that was formed in the last set of reactions. So these energized molecules travel across the cell to where the Calvin cycle is going to happen. Those molecules will then be broken down again, and their energy will be used to build glucose. So ATP is going to get broken back down into ADP, and NADPH is going to get broken back down into NADP+. Let's think about those specific reactions in a little more detail. What kind of a reaction is it when ATP is changed into ADP? Well, that, of course, is a dephosphorylation. A phosphate group is removed. What about the other reaction? NADPH changes back into NADP+. Well, the plus sign here tells you that this molecule has lost some electrons. So NADPH is going to be oxidized into NADP+. It will release its energized electrons into the reactions of the Calvin cycle. Now we need some raw materials to build our glucose out of. Where do those come from? Well, that's where good old CO2 comes in. Remember that CO2 was a waste product in cellular respiration. Here it's a necessary reactant. 6 of these little CO2 molecules are going to be needed to build glucose because if you remember, glucose contains 6 carbons. It's C6H12O6. Those depleted energy carriers that we end up with after this reaction are then going to travel back to the light reactions and start this cycle over again. The enzymes that catalyze the dark reactions, also known as the light-independent reactions, are out in the stroma of the chloroplast. So we're not on the thylakoids anymore. Now we're out here in this thick liquid. What we're actually trying to make through the synthesis part of photosynthesis are sugars. Now we're not going to start out by building the big sugars. We need to build small sugars first, and then we can use those little sugars to make the bigger ones. The sugar that's actually going to be produced through the Calvin cycle is this one, glyceraldehyde 3-phosphate. It's okay if you just know it by its abbreviation, G3P. G3P is a 3-carbon sugar, so there would be one there, there, and there. And it can be used as a building block, almost as a monomer, to build the big sugars like glucose. Glucose, if you remember, has 6 carbons in its chain. We would need 2 glyceraldehydes to make one of these glucose molecules. The Calvin cycle, or the Calvin-Benson cycle, sometimes also called the C3 cycle, is anabolic in nature. So it is endergonic. It's a building type of reaction that requires an input of energy. In order to make our one little sugar, our one little G3P, we're going to require 3 CO2s. So the CO2s are actually going to enter this cycle in groups of 3. The Calvin cycle may look a little bit like the Krebs cycle to you that we studied back in Chapter 9, but there are a few important distinctions. First, the Calvin cycle is anabolic in nature. It's endergonic. So what we're trying to do is build something. Specifically, we're trying to build this little sugar, and we're going to have to take in some energy from energy carriers to do that. The Krebs cycle was exergonic or catabolic. In that reaction, we were taking acetyl-CoA and breaking it down a little at a time and pulling that energy out and putting it into energy carrier molecules. The other important distinction is that in the Krebs cycle, we had one molecule entering at a time. One acetyl-CoA would go in, go through the steps, and be finished. In the Calvin cycle, though, we have groups of molecules entering all at once. For instance, we start with three CO2s, and they end up making three of these molecules, and then end up making six of these molecules, for instance. So we have groups of molecules interacting with one another in each step. The first phase of the Calvin cycle is known as carbon fixation. This is the step in which we're going to capture our little carbons in the form of three CO2s. So three CO2s are going to go in. Now, just as in the Krebs cycle, we need to have some pre-existing molecule to stick them to add them to. And that molecule for us is going to be RUBP. RUBP consists of 5 carbons each. So we're going to stick a little carbon from carbon dioxide onto this 5 carbon molecule, and we're going to temporarily make a molecule that has 6 carbons in it. So 5 plus 1 makes 6, and there's 3 of those because we had 3 CO2s going in. Now, in the second part of this phase, those 6 carbon molecules are going to get snapped in half into this molecule, which is just called PGA. You can just know it by its abbreviation. Now, because we had 3 6-carbon molecules, when we split those in half, we end up making 6 3-carbon molecules. So the number of carbons still adds up. Now that we've added our 3 carbon atoms to this cycle, in other words, we've fixed our 3 carbon atoms to other molecules, we can now take those molecules into phase 2 of the cycle, which is known as reduction. What's going to get reduced? Well, the thing that's going to be reduced are the 6 PGA molecules that we ended up with at the end of phase 1. Without going into a lot of detail, those PGA molecules are going to get converted and changed around into our 6 little sugars, 6G3Ps. Now, this process is endergonic. It requires energy. So that's where our little energy molecules that we filled up through their light reactions are going to come in. 6 of our ATPs are going to come along, and they are going to drop off 6 phosphates. They're going to add them to those PGAs, and it's going to make this intermediate product. The ATPs are dephosphorylated and now have to go back to the light reactions and get refilled. We're also going to use up 6 of our NADPHs from the light reactions here. They're going to get broken down into 6 NADP pluses. They're going to drop off some of their energized electrons, and that's going to provide the energy to convert this intermediate into 6 of our little G3P sugars. So we made it. We're finally at the sugar step. That was the point of the synthesis reactions. At this point, you may be thinking, fantastic, we made 6 sugars. This was a really profitable little cycle. Well, I've got some bad news for you. We're not going to be able to take away and use all 6 of these little sugars that we made. Why is that? Why can't we use them all? Well, if we just wanted this set of reactions to occur once and make those 6 little sugars, sure, we could take them all and use them to make glucose and sucrose and starch and other things. But we want this process to be a cycle. We want it to happen over and over and over again so we can continually make sugars, so we can continually make these bigger products. So let's do a little math. The cycle began with 3 of these molecules called RUBPs. Each one of those RUBPs contained 5 carbons. So 5 times 3 gives us 15 carbons total. So we need 15 carbons to start this cycle. Now, let's look at the sugars that we have. We have 6 of these sugars, and each contains 3 carbons. 6 times 3 means we have 18 carbons to play with. That means that we can take away one of these sugars and use it for other things, but we really need the remaining 5 to go back into the cycle so that we can start this process over again. So the profit from this cycle is one little tiny sugar, half a glucose, essentially. And the remaining 5 sugars go back into the cycle to start it again. Phase 3 of the Calvin cycle is going to allow it to start over again. It's known as the recycling of RUBP or the regeneration of RUBP. So what we're going to do in this phase is take those 5 3-carbon sugars, those little sugars that we have left over, and we're going to rearrange them back into RUBP. Each RUBP contains 5 carbons, so we're going to go from 5 3-carbon sugars to 3 5-carbon sugars. This requires several steps, and it is endergonic. It does require some energy. So 3 more ATPs are going to get broken down in this phase. They're going to be dephosphorylated, and that energy is going to be used to reform our RUBP. At this point, those RUBPs can grab some more CO2, and it can start the cycle over again. 3 of those guys. I mentioned that this Calvin cycle is anabolic or endergonic. So how much did it cost us to make one little tiny sugar? Well, if you add up all of the different steps and all of the different costs, you'll find that it cost us 9 ATPs and 6 NADPHs to make one little G3P. Remember that to make a glucose molecule, we actually need 2 of these G3Ps. So total, it would take us 18 ATPs and 12 NADPHs to build one glucose molecule. That's a pretty energy-hungry process. Congratulations, you guys. You're finished. You made it to the end of this very dense, very complex chapter. Bob Ross would be proud of you. Make sure that you thoroughly fill out your notes and give each of those practices, those your turns, a try. Bring them to class with you, and we'll discuss them and work our way through that material so that hopefully it makes sense. Thank you very much.
Plants provide us with food to eat and oxygen to breathe. They perform this amazing feat by the process of photosynthesis. Let's take a closer look. Photosynthesis requires carbon dioxide, which diffuses into the leaf through small pores and then enters the cells. Inside the cell, carbon dioxide diffuses into the chloroplasts, where photosynthesis takes place. Chloroplasts use energy from light to transform carbon dioxide and water into sugar and oxygen. Zooming into a chloroplast, we see these flattened membranous sacs called thylakoids. Here, light energy is converted to chemical energy in the first phase of photosynthesis, the light reactions. The 2 green structures you see here are photosystems, large complexes of proteins and chlorophyll that capture light energy. An electron transport chain connects the 2 photosystems. Notice the small mobile electron carriers that shuttle electrons from one large complex to another. Now let's take a closer look at the steps of the light reactions. The photosystem on the left absorbs light energy, exciting electrons that enter the electron transport chain. Electrons are replaced with electrons stripped from water, creating oxygen as a byproduct. The energized electrons flow down the electron transport chain, releasing energy that is used to pump hydrogen ions, the blue balls, into the thylakoid. In the photosystem on the right, light energy excites electrons, and this time the electrons are captured by an electron carrier molecule, NADPH. The high concentration of hydrogen ions inside the thylakoid powers ATP synthase, producing ATPs. The light reactions in the thylakoid have produced 2 energy products, ATP and NADPH, that will now power the production of sugar in the Calvin cycle. The Calvin cycle takes place outside the thylakoids in the stroma, the thick fluid of the chloroplast. At the beginning of the cycle, carbon dioxide molecules combine with molecules called RUBP. The resulting molecules go through a series of reactions powered by ATP and NADPH from the light reactions. Sugar molecules known as G3Ps are produced. Most of the G3Ps are rearranged back into RUBPs that will begin the Calvin cycle again. But the important product of photosynthesis is the remaining G3P sugar. Some G3Ps are used to build glucose, which can combine into starch or cellulose. Still other G3Ps form sucrose. And some of the sugar is broken down by cellular respiration using oxygen in the plant's own mitochondria, generating ATPs that can power other work of the plant. Excess oxygen diffuses out of the leaf through the pores, while more carbon dioxide enters. With 3 simple ingredients, carbon dioxide, water, and light, plants produce sugar and oxygen by photosynthesis, powering plant metabolism and ultimately providing your fuel as well.